Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief, Medical Product Outsourcing, and moderator for today's program. You've logged on to the MPO webinar, Big Changes to ISO 10993-1. What is happening to the main biocompatibility standard now? Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today, once the webinar has ended, or if you'd like to forward it to a colleague, this session will be archived for a year. The archive will be able to be accessed through the MPO website, which is at mpomag.com. Also, we will be accepting questions throughout the presentation. You can type your question into the Q&A box in the webcast window. We'll collect all questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce today's speaker, I'd like to take this opportunity to thank our sponsor, Nelson Labs, a Sotera Health company. Nelson Labs is an industry-leading global provider of laboratory testing and expert advisory services. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, protective barriers, and tissue industries. Those at Nelson Labs know that every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now let's get started. Today we're joined by Dr. Helin Raagel, Senior Biocompatibility Expert at Nelson Labs. Dr. Raagel is an expert in biocompatibility, especially with respect to the relationship between medical device materials, biological systems, and the analytical procedures used to evaluate biocompatibility in vitro and in vivo. She has over 10 years of experience in scientific research, over 2 years of medical device experience managing validation work for regulatory acceptance, and is currently working as a biocompatibility expert in the technical consulting group at Nelson Labs, giving guidance and recommendations for biocompatibility assessments targeted for regulatory submissions all over the world. Now we'll begin with today's presentation. Dr. Raagel?
Hi, everyone. Thank you very much for joining into this webinar. If you know me at all, you know that I love to talk about biocompatibility and also ISO 10993-1 because they kind of go hand in hand, right? So basically, we're here to talk about my favorite subject. So I hope you are all settled in and we can start this discussion of what ISO 10993-1 is and where is it going. And because it's changing, what is happening there again? Exciting stuff. But before we go into the topic, I wanted to kind of show you the timeline, the evolution of ISO 10993-1 over the years. So to the far left side, we have the dark ages. That's joke, but really that's something that we don't really talk about. There has been previous ISO 10993-1 documents like the 1997 one, and there was a 2003 one, but it's old, it's collecting dust somewhere, so I think we can all agree we shouldn't go deeper into those. What we will talk about is ISO 10993-1 2009, which I will refer to in this webinar as the past approach. And we talk about this one because sometimes when I look at sponsor documents, I still see that revision pop up. And that's why it's important to talk about like where we were and then where we're going from there. So next, I put on a timeline this FDA guidance document. I know it doesn't have anything to do with ISO itself. However, it did have an impact on the revision from the 2009. And we will talk about that a little bit further later on. And what we can say right now is really with the 2016, there was a really big discrepancy of what 2009 version of ISO 10993-1 wanted from the testing side or assessment side and what the FDA guidance said. So now what was then taken into consideration, this FDA guidance, they tried to more align and harmonize them. So then there was this ISO 10993-1 2018 revision that was released that kind of harmonized a little bit. There was more that happened, which we will talk about further in detail. But so that's the current one. So right now in this webinar, I will refer to that as the current guidance. Also on this timeline, I wanted to point out that there was, which I always get many questions about, there was also an EN ISO 10993-1 2020. And people are like, well, what about this 2020 version, right? Well, I can tell you right now that that was basically the EU side harmonizing it for their evaluations. And as it says in that 2020 version, that normative text, the main text from the guidance of the 2018 version was basically taken over without edits. So we're not going to talk about that anymore. And then we have this new draft. And so do you want this little spoiler alert? I can tell you. So they took this 2018 version, and what they did is they throw it out the window. Okay, so I hear a lot of gasps happening. And when I read the first, the draft of the new 10993-1, I was also like, whoa, bold move. But we will talk about it a little bit further down the line. So what we wanted, what they wanted to do with it, and where do we go from there? Okay, so I hope everybody's on, the edge of their seats. So what we will talk about today is how the changes in 10993-1 have occurred over time, the 2009, the past approach, the 2018, the current approach, and then the new draft. So how it has shifted, what has shifted, do we see anything that is popping up that is really necessary for you to understand? And then we will talk about general global acceptance of these proposed changes in the new draft. And I can, give you another spoiler alert that there have been some negative votes. And we will talk about those too, like what are the negative votes, how they affect, and what is going to be coming up next to kind of make sure that we get to a point where we have one document that is both useful and also accepted around the world. Okay, before we go into the standard, the 10993-1, perhaps we need to kind of really just cover briefly why do we even talk about biocompatibility, and why is it so important that we do these assessments for medical devices? So why do we care? So I always say, okay, in my mind, medical device manufacturers, they have a good intent. They want to make a device that helps the patient to treat some disease, maybe enhance their quality of life somehow. So they have a very good intent. But as we all know, sometimes the good intent doesn't really translate to the best outcome or the best result. And I always try to kind of think about how to visualize it. And then, I remember, when I was a kid and I wanted to do something nice for my mom because she's great. And I always had the great intent in mind, but sometimes it didn't come out as I wanted to. So can you imagine, let's say a teenager is trying to bake some cookies for the mom to show that she appreciates her. And so the mom leaves and she's like, okay, I have this great idea. And then 2 hours later, let's say, mom comes back, the room is full of smoke. There's, in the kitchen, the dough is dripping between the countertop and the oven to the place where you can't quite reach to clean it. And then there's a big burn mark on the kitchen table. Okay, I'm not saying, can you see it? Can you see it? I'm not saying that this happens all the time, but we have to make sure that we have some sort of system in place to prevent that from happening, because we're not talking about a ruined kitchen table, we're talking about patient lives, right? So what we really want is that there has to be really clear instructions on how to make a device and how to use it, and then also have quality steps and verification that this thing wouldn't happen in real life, right? Another thing to think about is that what can go wrong with my device and how do we mitigate those risks? So we talked about the manufacturing process, how things could potentially go wrong in there. However, there's also down the line, what is happening during use? Can we see that there is something that potentially could happen, like wear or degradation, that we have to really understand and then mitigate those risks as well? Another thing that biocompatibility looks at, and right now maybe it's really, really popping up as an important question, is that if something happens, like a change, right, like a supply chain issues that we see oftentimes right now, how do we take that change and evaluate the medical device so that it still remains or continues to have a good biocompatibility profile for its intended use? So all of these questions are kind of wrapped under the term biocompatibility. And ISO 10993-1 is kind of like the standard, the Bible that covers the framework how to assess these, right? So let's talk about the standard now. As I said, we use the past approach, current approach, and then the future. So in the past approach, what we had was that we had a device, we had to define its contact type and duration of contact, and then we had to perform the test based on the famous or infamous table A1, right? And as you can see from the table, there's a lot of X's there, lots and lots of X's, which basically meant that you just, got to do the test that is indicated in the table. And then because of that, this old school approach is called the checkbox approach, right? So it's a checkbox approach. So if you show, that these boxes have been checked with the testing, you're good to go. But the biggest downside to this approach in the past was that people didn't really focus on understanding how their devices are made, how do they interact with the patient. There was no upfront discussion about what goes into making up this device, right? How do you pick the appropriate materials and processing for your device? So it was really like, if the test passed, then you're good. But we've sometimes seen that this approach maybe, first of all, it might not be the best way to do it, right? And sometimes it doesn't capture the real concerns, right? And then with the ever increasing complexity of these medical devices, we really are seeing that this checkbox approach is not the right way to go. And also, if we're doing all of these animal testing for devices that really should be just evaluated, then why are we using all of these test animals in these preclinical studies that really don't need to be used, right? So that is why the 2018 version was released, to give it more like a flexible guidance to evaluations, where the goal was really to look at the risks of the device and define the appropriate testing approach that could be used for your device, right? So there was no more like this cookie cut approach there. So the checkbox thing, I think we should move past. It's not the right way to go. Okay, so the current approach, right? The 2018, everybody has seen this table A1, again, the infamous table. And as you can see, with the 2018 version, this table, expanded drastically, right? There were new endpoints added. And I might have to say, it's not even the whole table, because the whole table did not fit on the slide. That's how big it was. So yeah, okay, so we have different endpoints being added. Mostly they were adding these systemic toxicity endpoints, these long-term systemic toxicity endpoints. For example, we have the chronic toxicity that was added. We have carcinogenicity. There's also the reproductive toxicity and developmental toxicity that was added that's very specific to very specific devices, right? Degradation was added as a point for consideration, right? Another thing that was changed was this physical and or chemical information. Okay, so now this one was really added to hone in on that thing where I said, oh, before we didn't really look at the materials and processing. Now this one really says, okay, before you do anything, you've got to look at the physical information and the materials that you're using, the available information, that then you can decide what happens next based on that information and, you know, the device intended use. This column also refers to the so-called extractables testing with the chemical characterization approach. I do have to say, it really needs to be considered whether you need to add chemical characterization or not to your device. I'm a firm believer that for the, specific devices that, for example, do not have systemic exposure or have a very limited contact, you should not go down that path. But you just have to consider it, right? Another thing that was added was the better alignment with the FDA guidance document. Remember, I kind of talked about it, that the 2009 version of the ISO document did not really have the same endpoints that the 2018 version had. So the 2016 FDA, so that people were like, okay, if I'm submitting my device to Europe, for example, I need to follow the ISO. But then if I need to also, the same device, if I need to submit to the FDA, to the US market, then I need to add additional endpoints. And that really was hard for people because you had different sets of data that you need to show. So with this revision, and with the past years specifically, we've seen a lot of this trying to harmonize between the different documents, which is great because, in my opinion, the document really talks about the risks that the medical device has, and it shouldn't be dependent on where you submit, right, or who the patient is. It really should be the framework that gives you how to assess your medical device for its intended use based on what you know about it and what it's intended to do. So I think that's great. The work for harmonization has been great. I do want to give credit to everyone who's been involved with that. But the main thing in the 2018, as I mentioned, is to take a more holistic review of your device. It's not a checkbox approach anymore. You want to really take a look at your device. What does it do? What materials does it contain? Processing that is being done to it, and then really look at what gaps do we have in the available information that need to be covered with additional testing. And so this is really what the ISO 10993-1 is trying to say, okay? And as you can see, this evaluation of this risk-based approach that I love is really honed in with the E's in this big table, right? Where it says, E means that you have to evaluate for risk. So there's no more requirement for testing anymore, like that you have to do the test. If you have available data that covers that endpoint, you should definitely use that before you start testing again. So yeah. Another thing that was added was that the Annex B was swapped to bring in that the guidance on how to conduct a biological evaluation using this risk-based approach. It was brought into the document, and I think that really hones in on that. You really need to think about what is relevant and how should you cover those endpoints that are concerning with either available data or additional testing. Okay. And then because of this, because it's now more like, it's not just a checkbox approach, right? Now we have this view of like, let's take a look at the device, see how it's used, what is available information, and then you define the set of tests if needed. And that really drives home the message that you actually do need to start with a biological evaluation plan, right? The BEP. So that has also been brought into the standards. So before you start any testing, you should have this document that clearly states what you should do moving forward. And the BEP should capture all available information based on the device, its intended use and available information, and then define the testing. So to sum up, the current 2018 version, the current version, we have, this revised NXA with the new table A1, new endpoints, better alignment with the FDA guidance document. There's the emphasis on having the initial materials information, which is the only thing that is required for the plan or the risk-based approach for your device. And then that everything else is noted as with an E. That means that it requires evaluation, but that doesn't necessarily mean that testing is needed. There were also some new definitions added. For example, there was the transitory contact. And that transitory contact basically means that any contact that is less than 1 minute. And the idea behind is that if something has a very short-term contact to the patient, then most likely it doesn't really have time to start extracting something out of the device or the material. And therefore, maybe the way to, just do the testing might not be the appropriate way, especially when you consider the standard testing parameters. When you think about, okay, 1 minute clinical use contact versus, let's say, 72 hours of extraction at elevated temperatures for all of these in vivo tests, right? So that's something that was brought up, and I think that's really good. Really hones in on like that risk-based approach. And obviously the biological evaluation plans that everybody really should be, before you go into testing, really think about what is necessary and how we can address that. Okay, so now what? Okay, so the new draft, as I said, it's a complete rewrite. When we talk about, transitioning from the 2009 to the 2018, I'm going to say we've done lots of gap assessments, lots of gap assessments. And now I can see like, okay, there's this new draft coming out, sometime in the future. That's going to continue to happen. But first of all, I want to say, just to kind of like clear everybody's, mind and, take down the beating heart, racing hearts. The new draft, while it's going to be very different, we want to make sure that we understand that actually this risk-based approach is still there. So I'm just going to try to make you calm down here. Okay, so what I think is happening in this new draft, as I said, they scrapped it, the old version and started new, is that when we look at the 2018 version, right, I sometimes feel we're still stuck in the checkbox world, and I still feel like I'm pushing that cube, right? Pushing it. Why am I pushing it? Because it really says risk-based approach, and we really want to make sure that the new draft, the 10993-1, helps us in this process to make it more smoother, right? So I think that really is the key point to highlight with the new draft of the 10993-1. Okay, so another way to, really drive home the message here with the changes between the years and versions of 10993-1 is that, okay, when we look at the 2009 version, okay, so we have this, this large vehicle, it's a Toyota, so we know what to expect. Okay, it was like, okay, do the testing, we knew what to expect. Now with the 2018, we used the same base of the framework, right? We still, we added some stuff into it, let's say we added a better motor to make it more efficient, maybe added some airbags to make it more safer. But really the framework was like the overall document still had lots of that 2009 flavor in there. So I feel like we're still kind of held back by that weight of it. So it's just, we can't get maximum out of this vehicle or this document because we have such weight from the behind. It's just weighing us down. So what they're now trying to do is basically reinvent everything. It was like destruct and rebuild type of approach where you have a new framework, like in the disk-based approach still, but put into new frame and the new motor to hopefully make it so that it's really targeted to the risk-based approach. And we don't get this background noise of like falling back to that checkbox mindset again. So when we talk about the new draft, there is even more focus on this risk-based approach. They're trying to pull the ISO 14971 that talks about the risk management framework in general. So they're trying to pull that into ISO 10993-1. So that's really good. That really, brings in that, okay, you really need to know where to pull the right information, what information can you use, and really sets the stage for a risk-based assessment. And then we also have this big focus on the 3 Rs. We have already seen this come through with ISO 10993-23, the in vitro, the irritation one, where they really focus on the in vitro methods above the animal testing. So we have validated it. There was a long 10 years long round-robin study, and then trying to get this data out there to demonstrate that the in vitro method really works as well as, or even better, as the animal system. So now it's been written into the standard, and now this has been really the push forward, okay, we want to include this also in the 10993-1 to really move away from animal testing to find alternative techniques, and perhaps we can reduce the number of animals by, having studies where there's multiple things that we can look at the same time. So we get the same type of data, but from fewer animals. So this has really been honed in. Another big thing, again, the real big emphasis on the biological evaluation plans, appropriate description, because really it always starts with the clear description of the device. Then looking at the materials that go in there, processing. There's big emphasis now also on a continuous review of devices after they've been released to the post-market surveillance, to have technical data files on your device that expand from manufacturing to also the post during the clinical use. What's happening? Do we see something happening that we need to follow up with additional evaluation? Like, for example, wear, right? So that's something that is really starting to be put into this EP. So it's not just okay, one and done, we're on the market, that's great, but really the emphasis of putting the how it actually works in the patients, and do we see any concerns rising from there? That's also now being put into the document. And there's also, they were trying to put more guidance on how to write these biological evaluation plans altogether, because that was kind of lacking from the 2018 version. So now they're trying to build in more a more set framework so that everybody understands how to do it and why it's important. Okay, so basically when we take this this term, physical and chemical information, that really, that definition, I think, captures the BEP, right? It basically says we need to know the material, right? We need to know the manufacturing processes, and we need to know the type and contact during use, right? And then altogether, that knowledge that we have of all of these things need to be kind of combined together to define if we need any additional data to demonstrate that the device is safe for its intended use. And that really is what a BP is, right? So that's, I think that's going to be more highlighted in that new document. Another thing that is changing is the table A1. We have this long, extensive table, but when you actually take a look at it, right, it starts to become very repetitive. There's a lot of overlap between different categories. Like, for example, we know every single medical device that has any contact, indirect or direct, to the patient, they have to have these big three assessed, right? Cytotoxicity, irritation, sensitization, right? So do we really need to repeat that all over again, with like so many lines? And then when you also look at the different other endpoints, right, there's a lot of overlap between different categorizations, but the endpoints are very similar, right? So again, like, do we really need to have this extensive table that has so many different lines that you kind of like have to take a ruler to see, okay, am I like reading the right E's for my evaluation? So maybe we can present this in a different way, right? So they're really trying to see if we can make a new table that doesn't have this repetitiveness in there, but right now it's in Annex A, right? But now they want to move it into the main text, because that's where it really should sit, because that's like the where it talks about this is really important. So it shouldn't be like maybe hidden away behind doors like in the annex. Maybe it really should be in the main text. Okay, another thing that we're seeing is becoming different is that in the 2018 version, we have the ISO 10993-1, and then it references each of these accompanying ISO standards that talk about more specifically about the individual tests that are being done. But now the idea is perhaps into this new draft, we can put little snippets or like a summary of each of these tests into the annex of the draft. And the reason behind it is that we've seen a lot that some companies only buy this 10993-1. So they have this one document that they follow, that they really want to make sure that, okay, we understand the concept of the risk-based approach and what do we need. So they don't want to really spend money on buying the individual standards for the test. So then they can use this and at least get an overview of like what the test says and does, right? So, I mean, I guess it makes sense. So the draft has already been undergone some voting, right? And there was lots of feedback on the draft. More than 100 pages of comments were received, okay? And as I said, there were, some negative votes. Overall, I think in general, it was pretty well received, but there were two big negative votes. One from the US, which is basically, the US FDA, and Germany, which I would say is too soon, and other, notified bodies as well. But these two players, FDA and too soon, I mean, we need to make sure that these big players are okay with this draft, right? Because if we cannot get this standard to be, widely accepted, then all of these years of harmonization and moving towards one way of thinking is going to be gone, right? We're going to be back in the wild, wild west. where, the medical device manufacturers would be like, I don't know what to do. I'm going to submit to the FDA. Do I need to do this thing? I'm going to submit also the same device into, Japan or EU. And then you need a completely different set of assessments. And that's just, can we all agree that's not the best use of everybody's time and money? So I have to say, the draft is currently in a position where it cannot be withdrawn. Okay, so we have to work with this draft. So really what we need to do is to make the draft in a way that everybody can agree on, right? And so the decision was to form a writing team that really works on these problem areas, like what are the main concerns and how can we improve or incorporate that into the draft so that that everyone can agree on. And I have to say that despite of these negative votes, the players are actively being involved in figuring this out. So that gives me hope. And I feel like that's a good sign. So we're, there's work being done behind the scenes that really focuses on making this draft into a form that will be usable and accepted. Okay, so let's talk about a little bit about the main issues. Okay, so both from the US and the Germany side, we've heard that they do not like these annexes, like the annex, including the summaries of these ISO 10993 testing standards. And the reason for that is that when you have a very short description, right? I mean, if it's a short description, we can't put the entire standard. I mean, you would have like 1,000-page thing. So we have only the recap, right? But if you only read the recap, there cannot be really room to put details of the test or like some little things to consider. And because of that, some of the people that only read the little snippet, they might be misled about what the test does or how it should be done. And because of that, there could be misunderstandings that could result in either further or repeated testing, right? And that could also lead to, time consuming reviews and rejections. And that's really not what we want, right? Another thing from the US side that they don't really like is that push for in vitro methods, right? And we have seen that happen with the 10993-23 already, where we see, as I mentioned, there's a really strong emphasis of using the in vitro methods before the in vivo methods. And since the US FDA is still figuring it out whether they accept the in vitro irritation method or not. So then they have only partially recognized that the dash 23, because they only recognize the previous in vivo methods, but not the in vitro methods, right? But I do have to say, based on the conversations that I've had with people from the FDA, I know that they're actually working really hard on numerous in vitro methods and their developments. So I think they are being actively involved in this, and hopefully, we will overcome this in vivo versus in vitro dilemma. But right now, it's just, it is one point of friction here. The one thing that I also want to highlight is that when we have, let's say, the rabbit pyrogen test, right? And EU has strongly come out that they do not want to see rabbit pyrogen test. Soon it will be like out of their mix, right? But then now again, there's going to be this dilemma. Okay, do we do the rabbit because the FDA wants it, or do we do an in vitro method, like for example the monocyte activation test? It's like, do we do that instead, or do we have to do both? And that really needs to be figured out in the draft. Okay, so as we discussed, the current Table A1 is also undergoing changes. There's been an attempt to remove this repetitiveness, right? And really develop something that hones in on the fact that when we have a device, the more risks, the more invasive it becomes, the more risks it inherently starts to carry, right? And then we need to kind of figure out what would be the best way to have this, if it's in a table form, that people can really follow that thought process, right? So right now, what they have proposed is that none of the E's have changed. Like, you still need the same evaluation endpoints, but the way that the table is presented is completely different, right? But there also has been some discussion that maybe table for this isn't the best way to go. And I mean, I kind of agree, because when we look at the table, it basically pulls us back into that checkbox type of thinking again, which we don't want to do, right? And perhaps there's a better way to put it, right? And some of these endpoints, they don't even, become relevant for some devices. For example, like let's say we have a tubing, right, that provides some fluid to the body, right? It only has indirect contact, but if it's like used for, more than a limited contact time, then implantation becomes an endpoint for evaluation. But I mean, seriously, if it doesn't have any tissue contact, are we like direct tissue contact? Why are we even considering having an implantation test, right? Or that endpoint. So that's the reason why maybe we don't really want to have a table, and maybe instead we should have a flow chart, because I love flow charts. And so the idea would be to have it, the framework of the flow chart, in a way that goes over these questions, and then define, okay, what becomes relevant for your device, or the specific type of contact that you have, right? If it was up to me, I would propose that, first of all, like I said, everything starts with a clear description of the device. You need to really hone in on, like, how does it contact? What are the parts that contact? How long does it contact? And all of that. And then, we go into the flow chart. We'll say, okay, evaluate for the big three. Do we really need to do those tests? What do we have information-wise to cover those endpoints? And then the second question will be, is there any systemic contact? Okay, and if there is, right, then how long is the contact for? All right, and then you kind of consider the relevant systemic toxicity assessment approach, meaning what are the endpoints that are relevant? Like, are we just talking about acute? Are we talking about long-term contact? Also, like, can we, use the extractables analysis with the chemical characterization to cover, let's say, those long-term endpoints? Or maybe our device is just not suitable for that type of approach. Then what should we do to really address the concerns for that specific device, right? And then we can go further. It's like, is there any blood contact? Is it direct or indirect, if there is contact? And then really define the relevant hemocompatibility endpoints, right? And then you go further. Okay, is the device embedded in a tissue for a longer period of time, right? And then you'd be like, okay, do we need to consider implantation testing? And, if you have something that is permanent contact, it's an implant, stays there forever, obviously you do need to consider some. And then only then you go to that point, right? And then you also consider any special items that might be only for your specific device. For example, is it a degradable material, right? Do we need to consider different endpoints for assessment, right? And what those endpoints should be, right? Because that's really device dependent. Or if it's as a specific location. Do we want to, is it something that is implanted in the brain? Obviously, that's a very specific location. And then we need to kind of target those endpoints with the, let's say, the implantation testing. Also with the, like the chemical characterization, if we have something long-term in the brain, we need to consider neurotoxicity as well, right? But for other devices, we don't really need to consider that because that's not the endpoint of concern. So that's what I would propose, but we'll see. We're still, in the draft. We still have all of these kinks to figure out. But the main goal, is to have an ISO 10993-1 document that regulatory bodies around the world, including the FDA, will accept and recognize, right? But it would still incorporate more risk-based assessment, right? The risk-based approach, more emphasis on these in vitro alternatives, and the alignment with the ISO 14971, which kind of goes back to the risk-based approach, so that we really take a step back, we think about the device and what we know about it, and then define what testing needs to be done before we start any testing at all. Okay, so now I've done a lot of talking. Is there any questions that I can answer for you guys?
We definitely have questions, but thank you for that thorough and informative presentation. To everyone out there, once again, don't forget this presentation will be archived on mpomag.com for a year. Now, as Dr. Raagel indicated, we'll get to the Q&A. We'll try to get to as many as we can. There's been quite a few that came in. So the first one, and this was asked a few times, do you recommend, or I'm sorry, when will the new ISO 1093-1 be published, and is it possible to read the draft?
Yeah, so that's a great question. I also think okay, how should we prepare, right? So right now, since we're still in the mix of figuring it out, trying to make sure that everything has been answered, all of the really friction points have been smoothed over. I don't foresee it happening for another year at least. I think the more feasible answer is 2024, but that's where I kind of have to, just give my estimate, right? Because nobody knows. If I had the magic 8-ball, that would be great. But so I don't think the draft is, available for everyone. I can kind of see if there's option to share it, but I don't think it's really for everyone to read, at least for now. I'm sure like sometimes, like the FDA guidance, sometimes before they release it to the public to see kind of what they see. Maybe there's an option for that, but I'm not sure about that.
Okay. This question came in early. Do you recommend a component manufacturer to use ISO 10993 biocompatibility test, or is USP6 enough?
Yeah, so it really depends. The USP class 6, right? That only really looks at the 3 endpoints: irritation, acute systemic toxicity, and a short-term implantation study, right? When we look at that, the way those are run are very similar, or they can be very similar to the ISO ones. I feel like if you have something that has only a short-term contact, you can kind of go back and forth, right? But again, like if you have something that is long-term contact, then these endpoints are not going to be enough, right? When you talk about parts versus the final finished device, in the end, we still need to evaluate the final finished device. If you have a part that is put as such into the final device, I think that would be, really good to have that initial information on. But if you only have, material information, you want to give some kind of like reassurance that your material is good to use. But I also wouldn't, go much deeper than the USP class 6 if you have a material, just the material, right? So it really depends on how that part is used in the final finished device, because all of the notified bodies and regulatory bodies around the world will care about the final finished device. I hope that answered the question.
Okay. Well, the next question, with a transitory medical device with a coating material, do we require biocomp studies?
So it really depends. I would say for a transitory contact, again, like because the contact is so short, we really need to take a step back. Is it really needed, right? When I take a look at something, when it's made out of materials that we know, yeah, and even the coating, right? If it's already, it's a very, standard material, I wouldn't do it, just because the way that we run the tests are not really answering the question of this device's clinical use. But I do assure you, you have to have a document that covers that. So yeah, so you have to have a justification for not doing it if you have some coatings, right? But you can really think about, okay, what do we know about it, and what could be the concern? Because sometimes we even see, like, with even very short contact time, there could be there could be some chipping from the coating, right? That could be concerning. But if we can show that that's not a concern, I would really, really, really encourage you not to do the tests.
Okay, fantastic. Next question. Is there any potential for shorter extraction times for devices with limited use? For example, if a device has 10 minutes of contact, could it be extracted for 1 hour instead of 24?
Mm-hmm. Yeah. So I understand where you're coming from. And I've had this discussion many times. Again, like when we talk about risks from the clinical use, right? Then we do need to consider how the device is used. However, we know that the regulatory bodies are not going to go what is, beyond what is said in the standard. And for cytotoxicity, for example, it says no less than 4 hours, right? I mean, you can have investigational studies that do not really follow that to show it. That's fine, I think. But in the end, for the submission, you have to show what they want to see. That's just kind of like the sad reality right now.
Okay. With long-term deterioration, would that imply that we would need to reconsider doing BC tests in 2 times, fresh and aged by shelf life? Would working on an accelerated age sample cover all the lifetime of the medical device?
Mm-hmm. Yeah. So right now in 2018, it says like 1 line or 2 lines. Think about the entire life cycle, right? That's how much it is right now. We have seen that things do change over the course of their lifetime, right? And that has been like really spurring this conversation, like how much do we need to do up front, right? My stance is that we need to evaluate at one point. We do not want to do double unless it's absolutely necessary, right? So what I usually recommend to my clients is that you start from, the T0, and then you take a look at what happens to the device, right? Let's say it's supposed to sit there on the shelf for 2 years, right? And then like you said, you do the aging study. So you look at the age device, and do you see anything that is visibly changing? Like, is there, like sometimes we see like blooming, like color change on some tubes. So clearly something is happening, right? Then that's a concern. Or like sometimes, what I think, okay, from like catheters or like something that goes directly into the blood, this aging, we know that aging could cause like a little bit brittleness of some of the materials. So we really want to see, okay, do we see any difference on the surface that could come down to having an effect for, let's say, some hemocompatibility issues, right? So, but really, I think we should look at the device at the age state and see if there is any concern, right? I mean, as I said, I don't want you to do additional testing, but we always recommend also running, let's say, a comparative cytotoxicity test on the age device, so that kind of you can see, like, because it's so sensitive, anything that could potentially happen will happen there. As we all know, this test is, prone for failure. So if you have the T0 and the TH device, and you can see with the cytotoxicity that there's nothing changing and there's no obvious real changes occurring to the device by visual inspection, I think that's right now where we should stop. But then now with the new draft, the new draft really focuses also on this continuous review, right? So that you take a look at the clinical data from when you have something already submitted, and then supplement that into your biocompatibility package for your device, so that you do the due diligence or the vigilance to really assure that the biocompatibility profile is continuing to be safe for use.
Great. The next question: was anything covered in your presentation already binding? As I understand it, since ISO 10993-1 is in stage 20, all presented parts are still under discussion in the working draft, and consensus has not been reached.
Yes. So there is, I mean, I know I like very theatrically threw away the 2018 version, but there are, some parts of it that have been brought in back to the draft, maybe some slight verbiage changes. So the overall framework is not changed, right? That's really what I want to say. It's like the risk-based assessment, that is still there. So if you're doing it right now, just keep on doing that, and that's great, right? The way that it's presented and those figures and tables and flowcharts, those are still under discussion, right? Because we want to make sure that it is giving that flexibility for evaluations that really drives home that message of risk-based approach. But it's also not so vague that nobody knows what to do with it, So I think we're still kind of hashing some of these things out, and that's why it's going to take some time before it's released.
Okay. A lot of good questions. Is there going to be guidance on determining suitability of similar existing information before determining the need for additional animal testing?
Mm-hmm. Yeah, and that's a great question, because as I've already, 50 million times today, talked about, you need to consider all available information, right? So that's something I think that the draft really will help to define what testing data or what data from previous experience, or like some other information that you already have, how that can be incorporated into your risk assessment, and how much, like, this comparison of like what we know about the material versus the final finished device, that really needs to be honed in. And as I said, the draft really wants to give you a better guidance on how to conduct these biological evaluation plans that take into consider all of this. So I do think that it will be much clearer than it is right now in the new draft, in the new released version.
Okay, great. What about a device like a syringe where the medication to be injected is in touch with the plastic? Does this standard apply? If not, which standard does apply?
Mm-hmm. That's a great question, because I see these pre-filled syringes and I'm like, darn it. Because right now it really, so yes, the syringe itself is a medical device, right? But whatever is inside, that might be a medical device, might not be a medical device, right? And now it's kind of like really up in the air. What do we do with these, right? So that's why I feel like my proposed flow chart would be much easier to follow than, like, checking these E's in the table, because really the syringe is a very special case where you have only the needle contacting directly, and then you have like the syringe, right? And then it becomes like, okay, well, how do we assess, sometimes the syringe holds the whatever thing inside for a pretty long time. Are we concerned about what's coming out there, right? But it's only maybe acute exposure. How do we assess that? So this really is a special case. And I deal with that for some reason now, every week I see some of them pop up. So it is a very special case. In my opinion, you kind of have to, again, take a step back. Okay, what do we know about the materials of the syringe, right? Because if we know that the syringe has already been used for something else, we're not going to test for irritation sensitization again, right? And then it really matters, okay, the exposure, systemic exposure, what is inside the syringe, how much can it start to really extract, right? And then evaluate that. That's how I feel it should be done, but you're right, there is no clear guidance on that right now. So sorry about that.
Well, thank you very much, Dr. Raagel. Unfortunately, that is all the time we have for today's presentation. I'd like to thank our speaker, Dr. Helin Raagel, for her insights. I'd also like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience.